The cell cycle is a series of steps that cells go through in order to grow and reproduce by duplicating themselves. The cell we will illustrate throughout this project is a 2n equals 6 cell. What does that even mean? 2n equals 6 means the cell is diploid and has 6 chromosomes total, organized into 3 pairs of homologous chromosomes. The 2 means the cell has 2 sets of chromosomes, one set from each parent. The 6 means the total number of chromosomes in the cell is 6. And n works out to equal 3, which means there are 3 pairs of chromosomes. The cell cycle is comprised of 2 parts, the interphase and the M phase. The M phase is where the cell actually splits into 2 different cells, while the interphase acts as preparation for the M phase. The whole thing starts at G1, where G stands for growth. During this phase, the cell grows larger and prepares for replication. It continues producing proteins and organelles to support the creation of a new cell. Some cells don't like the idea of having kids and decide to not replicate. In this case, the cells leave the cell cycle to move to the G0 phase, where they do not continue growing and don't replicate. This prevents unnecessary cell division and maintains specialized functions of differentiated cells, such as muscle cells and neurons. After the G1 phase comes the S phase. The S stands for synthesis and means that the cell begins replicating its DNA. After this phase, the cell has 2 times the DNA that it started with, as the cell makes copies of its chromosomes. Also, centrioles begin to form at the poles of the cell. After the S phase comes G2. G2 is very similar to G1, and the cell grows a bit more as it checks itself to make sure everything is in perfect condition before it moves on to the M phase. After G2, the interphase is over, and the cell enters mitosis, or the M phase. Mitosis has 4 sections: prophase, metaphase, anaphase, and telophase. Mitosis starts with the prophase. Here, the chromatin, or loose DNA, condenses into visible chromosomes. Each chromosome consists of 2 sister chromatids joined at a centromere, and the nuclear envelope starts to dissolve, allowing the chromosomes to move freely in the cytoplasm. Next is metaphase. During this phase, the chromosomes of the cell line themselves perpendicular and equidistant to the centrioles of the cell. This ensures that each new cell will receive an identical set of chromosomes. The spindle fibers, or microtubules, connect to the centromeres of the chromosomes as the cell enters anaphase. Before the anaphase, the cell undergoes a spindle assembly checkpoint to ensure that all chromosomes are properly attached to the microtubules and aligned correctly. This step prevents errors in chromosome separation, reducing the risk of uneven chromosome distribution. Anaphase is where the spindle fibers separate the chromosomes at the centromeres, creating pairs of sister chromatids in both poles of the cell. As they approach those poles, you can begin to see the cleavage furrow forming, as you can see the cytoplasm of the cell begin to split. Telophase is where the cell begins forming 2 separate nuclei for the 2 daughter cells. It is basically undoing what the prophase did. The chromosomes begin uncoiling and the nuclear envelope begins reforming. Cytokinesis occurs after the nuclei are finished completely forming, and the cell fully splits into 2 daughter cells. These cells are exact copies of the original cell.
So bye guys. I hope you guys really liked it. The cell cycle, the end.